Okay, how many have heard about RNA polymerase and know its subunits and all that stuff in bacteria? You've heard of that, right, or not? Just briefly? You know what sigma does? It just recognizes a promoter. But that's, okay. Did you learn that in high school, or was that still third grade? Because that teaches stuff earlier and earlier. Okay, so I'm going to assume you've never heard of it before. And so that'll bore you more than normally. And then I'll bring you up to what I know about it. I've been following RNA polymerase all of my graduate school and professional career. In fact, I went to the University of Wisconsin as a postdoc from a PhD in plant molecular biology. I went to University of Wisconsin in some crazy, like 76 or something, just a little bit after discovery of DNA. And there was a guy on faculty there who, as a graduate student, and he was kind of a young faculty guy, as a graduate student, he discovered sigma, Richard Burgess. And so I would go in and talk with him on occasion, not very often. I was more interested, I guess, in the female postdoc he had. But anyway, they were different times back then. Anyway, I feel sort of this connection of some sort with RNA polymerase. So here we go. From here on out, it's about gene expression. We're through the recombination and repair and all these types of things. We're going to start with bacterial gene expression because the enzyme that we use was inherited from bacteria. We have the same enzyme at its core. And the Archaeobacter took a version of that and added subunits, and or perhaps the Eubacteria lost subunits. We don't really know. But anyway, we have the old-fashioned version of E. coli's RNA polymerase. It only has 4 or 5 subunits. Ours has 12 subunits, which we inherited almost amino acid per amino acid from Archaea. And so we'll try to see the big picture, and then we'll zoom in on some details as we go along. So this is in chapter 17 of Genes 12, and it takes about an hour to remove the blue. To remove the blue background from the slides, of course, takes about 6 seconds. But to remove all the shadows on the text and the shapes and all the group things, and then change the color of the font, it took me about an hour just to get part 1 this morning. So I apologize for not having the full series, but I've uploaded part 1. Here we go. Introduction, definitions. Coding strand, template strand. The RNA itself, if you were looking at the DNA, which of those 2 strands of the DNA represents base by base, except for the uracil-thymine conversion, which one represents the RNA, the messenger RNA? The template strand or the non-template strand? The non-template strand. And by convention, if you're just going to show the base, the DNA sequence of something, we only show 1 strand. We show the non-template strand. Why do we put such an emphasis on the non-template strand, where transcription puts 100% of the emphasis on the template strand? In fact, the non-template strand is completely useless. It doesn't participate at all in transcription. It's because of what DNA encodes. It's the messenger RNA that's important. How about for genes that don't code proteins? Ribosomal structural RNAs, tRNAs, all of that is represented by the non-coding strand of the DNA. By convention, if you see on the two strands going from left to right, it'll be the top strand, which will be reading 5' to 3' by convention. But you have to look at that as we go into the databases, as you probably found out. Sometimes that's flipped, and you have to pay attention to orientation that way. Okay, promoters. There are like three basic units in the gene. There's the promoter, there's the encoding portion, whether it's a protein or a structural RNA, and then there's the terminator. The terminator. Eukaryotes don't have a well-defined terminator, but bacteria certainly do. We'll learn about each of those components. The promoter, of course, it signals the place where the transcriptional apparatus should assemble and then initiate transcription. And you can see here a diagram of it. It's about, in a bacteria, it's about 75 bases. That's the average length of what a promoter looks like, but only small blocks within the 75 are actually conserved. We'll see those critical elements, promoter elements, if you will. So, direction, polarity. In the direction of transcription, away from the promoter, that direction is called downstream, right? If you're sitting at the start of transcription and you're away from the start, it's called upstream. So upstream and downstream, these are common things I assume you know about and have heard about before. So what's the structure in terms of, polycistronic or monocistronic? What's the difference? A polycistronic is how most messages are configured in prokaryotes. The genes normally, the vast, overwhelming majority, with just few exceptions, have no intervening sequences, no introns and things like that inserted in the middle of their messenger RNAs. And it's pretty much the rule, but not with lots of exceptions, that you'll have several coding regions all on the same transcript. In a eukaryote, it's not like that at all. You usually have introns. A perfectly good message can be interrupted 5, 10, 20 times, and with great long sequences. You remember we were talking about the human gene, the longest one, the CTN, Connecticut cadenacy AP2, CNTN AP2. It's like 1 million base pairs long, yet it codes a normal sized protein. That's because most of its sequences are introns. They get spliced out. You never see that, with just a few exceptions, in bacteria. And in a eukaryote, with few exceptions, it's a single gene per messenger RNA, monocistronic. So that has implications regarding translation, doesn't it, in the ribosome. In a prokaryote, the ribosome has to know how to recognize an internal start to transcription. Whereas in eukaryote, that's not a big priority. It just has to know where the beginning of the messenger RNA is. The first codon, or the first ATG it comes to, is usually the right one to start translation. There's some exceptions where there'll be a short open reading frame, and then you'll have the primary reading frame. But for the most part, it's the first ATG you come to in a eukaryotic messenger RNA that would be used for translation. So you don't need a Shine-Dorgano, you don't need a ribosome binding site, any of that stuff in a eukaryote. And that means that the ribosome all it needs to do is recognize the 5' end of the message and then just scan along until it finds ATG. So now it's a question of how do you recognize the 5' end, and secondly, 5' end of something that's supposed to be a message. It's the cap that's put on after transcription, the 7-methyl-G, extranucleotide methylated, on the 5' end that says this is the 5' end of a true messenger RNA, not a degradation fragment, not tRNA, not structural RNA. In bacteria, there's no capping because it's interrogating every RNA to see whether or not it has a Chandogano. So it's bouncing around the inside of messengers, scanning along. It doesn't go to the 5' end. It could go as easily to right in the middle of the messenger RNA and start scanning. And if it sees an ATG, it doesn't care. It's looking for a Chandogano, then an ATG. So there are different approaches to translation, depending on whether you're a polycistronic or a monocistronic organism. Transcription bubble. There's some nucleotide links that I sort of obsess on, and so that means you might see them on exam, and it's the length of the bubble. What do I mean by the bubble? It's the, what's this thing, well, you know, it has to be turned on. It's the length of the melted region that the polymerase is actually sitting on. There's always, you have to open up the two strands, and as the polymerase zooms along, it's continually opening up and closing it behind itself. At least enzymes do some of that, but mainly enzymes take care of the winding tension, but the enzyme itself opens up, melts the DNA, and this little melted region is called the transcriptional bubble, and it has a pretty constant size, 12 to 14 base pairs. Both of the number groups you need to know are right here on the slide. 12 to 14 is the length of the bubble, the part that's open, and how much is actually in an RNA-DNA duplex before the RNA, the new transcript, gets ripped away from its mother, the template strand. 8 to 9 base pairs. So 8 to 9 is in an RNA-DNA duplex, whereas 12 to 14 is actually melted to form a bubble. So you'll see that in the form of a multiple choice. Be sure you pay attention to whether I'm asking about the bubble or am I asking about the RNA-DNA hybrid. When it gets longer than this, the enzyme's going to be moving along and the 3' end will continue to grow. And when it reaches that 8 or 9 base pair length, the back end here, the 5' end, will get dislodged from the template and be directed towards an RNA exit pore. Something completely different than DNA polymerase would ever think of doing, right? DNA polymerase wants to make sure that the new strand that it just made will remain forever associated with the template. But RNA polymerase wants to make sure that it gets separated from the mother as soon as possible. So it has internal structures that act as sort of a wedge here to make sure that the 5' end is constantly being taken away from the complementary template strand. Okay, direction of transcription. So you can see that the RNA is made first at its 5' end because you're always adding nucleotides to the 3' hydroxyl. That's just the biochemistry of the reaction. The new nucleotides are being added here, so just by definition almost, it grows 5' to 3'. Of course, its template is oriented in the opposite direction. And here you see the magic numbers again and all the little code words we've learned. Coding strand, template strand, 12 to 14, 8 to 9, transcription bubble, upstream, downstream. Leaving an alpha phosphate. Okay, how fast does it go? Can you remember how fast DNA polymerase is able to polymerize? So it's like, how many nucleotides every second? 880 or something crazy like that. Look how slow and deliberate, thoughtful, if you might consider, that RNA polymerase proceeds. It's subject to all sorts of nuance, input, reaching a consensus by all its regulators. Transcription is regulated by proteins, predominantly. Proteins that stimulate it, called activators, and proteins that inhibit it, called repressors. But proteins interfere with the process or facilitate it. It's not a change in conformation of the polymerase itself. Regulation, like 99% of regulation of gene expression, happens at the stage of initiation. There are 3 stages in transcription, right? Initiation, elongation, termination. The initiation stage is where the regulation occurs, either positive or negative. If it's positive, it usually acts in a real simple-minded way. There are a couple of places on the holoenzyme that have binding sites or buttons to push. They're sticky regions that control proteins stick to. And that's the basis of stimulation. A control protein will bind to the promoter, get its feet all situated, and then it'll just reach up and stick itself to the polymerase. And now the polymerase tries to get away. It can't, just by thermal diffusion. It's tethered. And that simple act of tethering the polymerase ensures that even though it's kind of a lousy binding site, it can't go anywhere. It has many, many chances to make it work. If it was a lousy promoter and did not have the regulatory protein there to help it, it would bind and then be off on its way. But it's constrained, it's forced to sit in its room and eat its spinach, asparagus, or whatever it is, Brussels sprouts. You don't have a lot of degrees of freedom there. Eat that or no TV. It bounces around. I don't want to eat it. I don't want to eat it. Eat it. Oh, okay, I'm good. Eat it, bad. And then it's off. So that's how you stimulate transcription. How do you stop it? A real simple-minded mechanism in bacteria. You simply get in the way of the polymerase as it tries to interact with its promoter. You have sites called operators. That's what the site of a repressor element is called. Repressors bind operators. And these operators are inconveniently located right in the way of RNA polymerase, sometimes in front of it. Oh, I was here first, sorry. Or right overlapping the site of the promoter itself. But it's mainly some simple-minded way like that. Blocking, physically blocking, binding to the promoter or movement away from the promoter. Really simple, elemental ways to regulate transcription in bacteria. Oh, so yeah, this is the thing. So bacteria have no nucleus, right? It's like, it is a nucleus. It's a nucleus that's gained ribosomes. It's all in one, right? The nucleus, everything's all mixed up. And so translation has to happen in the same environment with transcription. So the ribosomes are right there. You're like a bunch of wolves or hungry dogs. They're waiting for somebody to throw them a transcript. They need to translate, make protein. When a transcript starts to emerge from the RNA polymerase, the ribosomes just scramble to look for Shandell-Garno. Come on, there's got to be one here somewhere. Like, yeah. And then they start translating while the RNA polymerase is still transcribing. The ribosomes jump on the message before it's even finished and start following the RNA polymerase, stalking it, making protein. And there'll be a line of them. 1, 2, 3 ribosomes will be going down the message, each making a nascent peptide, following the polymerase. So guess how fast they go? They go about the same speed as the RNA polymerase. There'd be no evolutionary advantage for them to really be fast. They'd always be catching up with the polymerase and just kind of looking like, what are we going to do now? And so the polymerase moves 40 to 50 nucleotides per second. Translation goes, it's 15 amino acids per second. How do you convert 15 amino acids per second? 3 nucleotides per amino acid, that's 45. Perfect. They're pretty much synchronized, the rate that the ribosome follows the RNA polymerase. But the RNA polymerase, of course, doesn't just smoothly proceed with messenger production. It starts and stops and shakes its head and backs up and it goes forward because it's, it's sensitive to nuance. It's looking for input. It's, after all, control of gene expression. You have to get it right. The DNA polymerase, one's job, make the genome as fast as you can. The polymerase, it turns out, is sensitive to its own transcript. As it comes out the back or out the side, the RNA exit pore of RNA polymerase, if there's any base complementary region that's inverted repeat in nature, it will snap back on itself and form a little hairpin. That little hairpin is outside of the RNA polymerase, but there's a binding pocket on the outside of the polymerase that it just slaps onto and acts to slow down the polymerase and cause it to pause until it can unstick itself from that thing it just made. These hairpins are forming all the time, and they cause the polymerase to have a very uneven movement. That characteristic of RNA polymerase, to be sensitive to the formation of hairpins that it just made, has been exploited as a termination tool. That's how it terminates. You engineer the transcript so that a hairpin is made and the polymerase is right on a sequence that is highly unstable. If the RNA is a U and the template is an A, deoxy-A uracil is the most unstable base pairing. And so just pause a little bit over this really unstable region, which is a stretch of A's in the template strand, is enough to make the whole thing fall apart. This RNA DNA duplex, this 8 to 9 base pair bubble, just melts apart and can't get back together and transcription's over. That's how you terminate in a bacteria. We don't know how it happens in eukaryotes. Sort of, but we're just learning still. Okay, so I've covered a lot of ground here. Maybe we can get through some slides to reflect that. And you see here that, the other thing that's happening in bacteria that's a little different from us and nematodes and cats and sparrows and bunny rabbits is RNases, stability of the messenger. In us, a messenger RNA could last a day, hours, sometimes more than one day. In a bacteria, the RNases there, the little Pac-Men, are constantly chewing away. The average half-life is about 1.5 minutes in E. coli for a messenger RNA. 1 to 3 minutes. lots of turnover. You make it, translate it, or, snooze you lose, because it'll be degraded in just a minute and a half. So it's very responsive to cues that influence gene expression. Okay. How fast does this happen? How fast do you start a new message? What's the fastest you could probably start a new message? It's at the speed I'm doing right now. 1 Mississippi, 2 Mississippi. About 1 every second is as fast as you can make it happen. The fastest genes on the planet, in terms of ability to reinitiate, are the ribosomal cisterns, those ribosomal promoters. They're the most active in all organisms, and they can initiate the fastest, and it still takes a second. Think how slow that is in terms of molecular speeds. Once a second. So what's the longest? What is the most inefficient, stupid promoter in E. coli? Who gets the retard award? Well, what kind of life, what kind of frequency should we expect? Probably not days, because E. coli is dividing every 30 minutes to an hour or so. It's the lac repressor. It initiates every 30 minutes. It's going, oh, is that a promoter? Nah, it can't be. Look, it's so lousy. This really, I got to be really desperate. You call that a promoter? 30 minutes, it hems and haws. It's just a ridiculous promoter. You don't need many lactose repressors. So, physical biochemist. Boy, you can't even talk to those guys. Their nose is so high up in the air. Biochemistry, physicists and biochemists. I mean, you're going like, you're hoping the clouds will part so you can see their face. Are they looking at you? Do they have their ear inclined to thee? They calculated, if you take hypothetically, a promoter's about 75 bases, how fast, just by diffusion, a polymerase could check out every base. Because a polymerase has no map. It has no eyes. It has no little tiny hands. All it has is temperature to animate it. Just bouncing around because average molecular motion, temperature. Excuse me. And finally, it bounces wrong orientation, upside down. How can it ever find a promoter? Just the rate of diffusion is very close to the rate at which you see the movement. In fact, it's right at the theoretical maximum, or the theory is a little bit faster than the theoretical maximum. And it was in well, it still is to some extent. It's like a puzzle. How can this be so efficient? It should take forever for that to happen. And so there are some hypotheses, all of which appear to contribute. And here you see them outlined here. There's sliding, transfer, and hopping. So the way to read this is sliding. This is how sliding works. Notice it slides either way. There's no, the electric slide. It just temperature moves it one way versus the other. Till it, I think this is a promoter. Intersegment transfer. It doesn't have to diffuse the entire length as if it was stretched out across the ballroom, because it's all folded. Then you can jump long distances. And then hopping. It binds and then goes nearby. It doesn't wander off into diffusion space. And somehow, through the magic of biophysics, it all works out and can be explained now without it violating the chemical and quantum laws of the universe. Sliding, intersegment transfer, and hoping or hopping. Look at that. Look how great I've lined up those boxes. That's what happens, believe it or not, when you try to change the background from blue to white, the slide gets angry. Sometimes you have to completely delete those little red things and do it over again. They get stuck in one configuration. You open them up, they go like right back again. You just go, yeah. So, let's look at the stages of how the polymerase approaches its job, gene expression, recognizing a promoter. So you have most DNA, most RNA polymerase is bound to DNA of some sort all the time. How many molecules of DNA Pol 1, polymerase Pol 1, are in a cell, bacterial cell, the repair enzyme? 400, right? How many copies of the replicase Pol 3, DNA Pol 3 are in a cell? 10 to 20. How many copies of RNA polymerase are there? 13,000. And most of them are bound to DNA of one sort or another. So let's look at the stages of how it finds a promoter. Core RNA polymerase is stored at nonspecific sites. Okay, core. Before we even get into the subunits, we got to talk about core. So let's have a brief segue into subunits so we can understand core. You have the largest subunit is beta prime. The second large is subunit, which is almost the same size as beta prime, is beta. And those two together look like pizza claws, like a crab or a lobster or something. And they grab the DNA. The active site is at the intersection of the two claws. There's a pocket there sitting and glowing in the dark, and I think it's purple if you look at all the slides. It's throbbing purple in there, a magnesium ion. But I thought polymerase had two magnesium ions. Yes, it does. The second one rides in on the deoxynucleotide. Not the deoxy, the ribonucleotide. This is transcription now. Okay. Beta, prime, beta, that's two. Then you have omega, which sits back here, which is involved in assembly and some other things. And then, so that's three, 1, 2, beta prime, beta, omega. And that's the core. Well, no, you got two more, don't you? You have alpha. Alpha. Alpha is one of the subunits designed to interact with control proteins. There are two copies of alpha. It's like the front part and the back part, the N-terminal part, we call that the front. It's connected to the main body of the RNA polymerase. And then you have a 19-base flexible, a 19 amino acid flexible linker. And then you have this little snowball on the end that's floating around called the C-terminus of alpha, which is there to either bind DNA and make it so the polymerase binds tighter to the promoter, or it will bind a control protein, which in turn is bound to DNA. That's the core. Sigma is the only thing that's left out of here, and sigma, its job is to bind just 25% of the time. 25% of the time in the life cycle of the polymerase, whether it's bound at stupid DNA, or it's bound at promoter, or whether it's elongating. Sigma is a transient partner, and when sigma binds, that's called holoenzyme, complete the whole. Okay, so the core, that's the stupid polymerase. It has no brains. The little Garmin insert with all the maps and stuff, that's sigma. It knows what a promoter looks like. The core polymerase can transcribe just doesn't know where to start, but if you put it on DNA, it'll start at any little nick and lots of random places. So the core polymerase has fairly high affinity for DNA. And you can see there in this figure, high affinity, very stable. It goes on fast and comes off slowly. When sigma and half of the DNA, half of the polymerase, what's that, 6,500 molecules, is always bound at non-promoter DNA very tightly and not going anyplace. It could take hours before it comes on, goes off. Its half-life is really long, just sitting around in reserve. When core binds sigma, when sigma finds a resting polymerase, it now changes the configuration of the polymerase, causing it to lose affinity by 10,000-fold for non-promoter DNA. At the same time, it increases affinity. It both increases and decreases affinity for DNA, just depending on what kind of DNA. It increases affinity for promoter DNA, but grossly, vastly decreases overall affinity. So now the polymerase can begin hopping like a little bunny, looking for a promoter, and doing a little slide, a hop slide, a hop intersegment transfer, because it's in an open configuration, because sigma is bound to it. There you see the 10,000-fold reduction. Increased affinity for promoters, 1,000-fold. Reducing stability of the loose complexes, stabilizing more frequently, stabilizing, okay. So, let's see here. So this is the place where we talk about open promoter, closed promoter, I presume. So what are the steps? It binds to double-stranded DNA, and it's called a closed promoter because the DNA is closed. The strands are not melted open. Binding the closed promoter is a reversible step. The next thing to happen is the strands melt and it becomes an open promoter. When it becomes an open promoter, for all practical purposes, it's considered to be irreversible. It will stay there until it transcribes. It will take a while, but it won't go back to a double-stranded configuration and then come off again. So the least stable is the double-strand closed promoter polymerase complex. The next stable association is open promoter, considered the first time in the series that it's irreversible. And the most stable interaction is called the ternary complex. Ternary meaning 3 components. It means it has protein and DNA, that's 2. And the third is RNA. And so it begins before it technically initiates, it's still making transcript. That sounds counterintuitive, doesn't it? It actually is transcribing 8 to 9 bases, but in the lingo of the biochemist, it's not formally considered elongating yet. It's still initiating because it cycles through 8 to 9 bases maybe a couple of hundred times before it actually can leave the promoter. That cycling, just making 8 or 9 bases and then stopping, 8 or 9 bases and stopping, is called abortive cycling. That is still part of the initiation phase. We'll see a little bit more about that. So the ternary stage is the most stable, and sometimes that appears on quizzes. But even beyond ternary, it's actually a subcategory of ternary. But the absolutely most stable phase is the elongating polymerase itself. Once it has left the promoter and truly in the elongation mode, that ternary complex is even more stable than the abortive cycling ternary complex. very little polymerase is free. What percentage is actually engaged in elongation of transcription? This is in one form or another exam. Here we go. This is, I'll skip ahead. This is the figure you should have in your mind when you try to answer that question. This little pie chart here has actually gotten a lot of people in medical school because they were just 1 point away, all their classmates. They didn't know this one question. This is, usually appears on the exam. I'm going to ask you something about the ratio of sigma to the rest of the subunits. You don't need as much sigma as the other subunits of RNA polymerase because sigma is only a transit association with the core. 50% of the enzymes are core resting versus core elongating. So half of it is just sitting at non-promoter DNA as core. 25% is bound to sigma, trying to find, or it's already found, but trying to initiate elongation sitting at a promoter. This is the only step where you need sigma. Once the polymerase leaves the promoter, in fact, sigma must leave. So there's about if this is 4 parts, 1, 2, 3, so it's like a 1 to 3 ratio, right? Of sigma to all the other subunits required. Let's go back. Okay, answer 25%. Let's get on to the topoisomerases. You have a gyrase, introduces negative supercoils in front of the RNA polymerase, and then topoisomerase removes negative supercoils following the RNA polymerase. We won't go into any of the enzymatic mechanisms of the topoisomerases, but that's you need to know. Gyrase precedes it. If you're opening the strands, you know, and moving and opening the strands, you're going to build up knots in front, supercoiled knots. They are constantly being relaxed by the gyrase in front, and then you're reforming the supercoils behind the enzyme using topoisomerase. Initiation, most of regulation, elongation, termination. These are the steps. Abortive cycling, 3,000 polymerases per cell. We've already talked about this stuff. The closed promoter complex is reversible. That would be up in this step here where you're binding non-promoter DNA. And then the open promoter complex where sigma is joined and it's actually found a promoter and melting it is the first irreversible step, then of course the ternary steps. Abortive initiation or abortive cycling, you see that it's a property of all of the RNA polymerases to sort of stutter at first before they get going, like spinning their wheels. Our polymerases do that also. Nobody fixed that along the way. Still there, doesn't seem to hurt anything. In fact, it's probably essential to the whole mechanism. So how does that work? How can you transcribe 8 bases and the polymerase doesn't move? Is it not impossible? How does it do 1 and 2 and 3 and 4 and the polymerase is just sitting at the promoter because that's what's happening in abortive cycling? It does it by a process called DNA scrunching. The template is coming into the active site and being sort of bent and warped and stored in the active site, packed in there for about 8 or 9 nucleotides. You can cram that many bases of template inside the enzyme before it explodes. And that determines the length of that little transcript. You start, get the first one, then you get the second one. That's the hardest step. Putting the first one and linking it to the second one. That's something DNA polymerase never learned. RNA polymerase needs no primer. It brings in the first one, connects it to the second one, then goes 1, 2, 3, 4, 5, 6, 7, 8, 9. And if it doesn't let go, it goes, and it springs out the front, the DNA, like a jack in the box. The little RNA escapes out the exit pore, and it starts again. 1, 2, 3, 4, 5, 6, 7, 8, 9. And if it can get that little RNA out the exit pore, 10, 11, 12, 13, 14, 15, it's chugging away, and it's in elongation mode. That happens 100, 200, 300 times every time poor RNA polymerase tries to start transcription, even in us. DNA scrunching. And the amount of DNA that you can pack inside in the scrunch, of course, you can see how it's related to the actual size of that little abortive transcript that floats away. What's the fastest promoter clearance time? Well, of course, once per second, ribosomal. Well, look at that. That little stupid box came first. Which came first, the slide or the box? Apparently the box. And this is all the stuff that we talked about. Here we go again, all the stuff we've saved ourselves. I hope a box comes floating in here. How long is the RNA-DNA hybrid? The RNA-DNA hybrid, not the bubble. I'm guessing 8 or 9. That's the bubble, and that's the RNA-DNA duplex. How much time? We got 1 minute? It's a wash, 2, 3, nobody cares. They stopped listening long ago. Okay, thanks for the little morale boost. We'll keep on going until people throw stuff, actually, like 30 seconds. Now we're going to look at the actual structure. That's an incredulous expression you have on your face. Like, really? He's going to keep us here? Even though you can see we all want to leave like crazy? I'm not receptive to people interactions. I'm not a people person. Phage. T7, they have very simplified RNA polymerases. One subunit, they combine it all into one big protein. So it's possible to do it. In fact, see this little thing at the end here? That is not a pink M&M, but that's a domain that is recognizing the T7 promoter. So it's possible to build it with just one thing. The length of the T7 promoter is like 6 or 8 nucleotides. You can clone it onto any gene you want to, and people do that in various biotechnology applications. It's a much speedier enzyme, 200 nucleotides per second. See, it is possible to transcribe much faster than E. coli really does it. It's slow for a reason, so it can be regulated. And I think this is a good stopping point. We'll talk about the thumb, the finger, the palm, and we'll go much faster on Thursday.